Back. So this video is specifically going to focus on our BOD experiment, our biochemical oxygen demand experiment in the water chapter. So the next thing we're going to look at is we're going to look at the BOD test. So before we begin, we need to figure out what does BOD mean. So BOD stands for biochemical oxygen demand. So for biochemical oxygen demand, it is the amount of dissolved oxygen consumed by biological action in a sample of water that is kept in the dark at 20 degrees for 5 days. And they're three really important pieces of information for exam questions. In the dark at 20 degrees for 5 days. So, to test the biochemical oxygen demand of water, we use a method called the Winkler method, and it's basically used to determine the amount of dissolved oxygen present in a sample of water. In this experiment, a water sample is collected in 2 bottles, and it's an experiment of 2 halves. The first bottle is tested straight away, while the second bottle is tested after 5 days when the bottle has been left at 20 degrees in the dark. The reason the second bottle is left in the dark is to prevent photosynthesis from occurring. It is also kept at a constant temperature, which is 20 degrees, as temperature affects the amount of oxygen present in water. Once you've tested both bottles, you can find the difference between your 2 titration readings, and that is the biochemical oxygen demand level. So this slide contains lots of exam question tips to help you. When you're filling the bottles of water, it's important that you fill the bottles under the water. So the bottles must be kept under water level, and that's to prevent trapping of atmospheric oxygen. If you're using polluted water, you may need to dilute your water solution to ensure that there is some oxygen left after the 5 days, because you don't want all of the oxygen to be gone out of your water in the 5 days. Your biochemical oxygen demand is measured in parts per million, and just be careful of the dilution factor when calculating if it is present in a question. And finally, the higher the biochemical oxygen demand, the more polluted the water is. So when you collect your water samples and before you do your titration, there's 3 chemicals that are added, and specific amounts of them are added. So 1 centimetre cubed of your manganese sulfate is added, along with 1 centimetre cubed of alkaline potassium iodide, and alkaline is very, very important here. When you add those, a white precipitate will originally form as the manganese reacts with the hydroxide in the water. This white precipitate will then react with the dissolved oxygen in the water to form a brown precipitate. Then, to finish off, what you do is you add 1 centimetre cubed of sulfuric acid, and this will cause the brown precipitate to dissolve, and the red-brown colour of iodine will now be observed in the bottle. It's important to note that each of these reactants must be added using a dropper, and the dropper must be placed underneath the level of the water in the bottle to ensure that they don't flow out. So just to make that last slide a little more clear, I've developed a flow chart here. So when we add the manganese sulfate and the alkaline potassium iodide, the manganese reacts with the hydroxide ions in the water, and that produces manganese hydroxide, which is our white precipitate. This then reacts with the oxygen in the water to produce manganese 3 hydroxide, and that gives us our brown precipitate in the water. Once the sulfuric acid is added, iodine is liberated, so we get I2. At this point, your bottle is now ready to be titrated against a standard solution of sodium thiosulfate, and it's very important to remember that your ratio for this calculation is a 1:4 ratio. So this slide here summarises everything that we've spoken about so far with our additions to our bottle through balanced equations. So the first equation here shows the brown precipitate formation, where the manganese hydroxide reacts with the oxygen to produce manganese 3 hydroxide. The second equation looks at the iodine formation, where we have our concentrated sulfuric acid allowing our iodine to be liberated. And this last equation here shows the reaction between the iodine and the thiosulfate, which will happen once you do your titration. That's it for this video. Make sure to revisit the learning intentions to make sure you understand everything that we've gone through.